Okay. So yeah, thank you so much for attending Mohonk Preserve's first webinar public program presentation. My name is Christy Bellardo. I'm the Education Coordinator for Public and Youth Programs here at the Mohonk Preserve. I will be monitoring and facilitating this presentation this afternoon. Before I start to introduce our presenter, Bill, I have just a little bit of housekeeping. We ask that you turn off your video and
fortunate.
On your audio modes, go ahead and turn them off. This prevents distraction to our presenter and others in the audience. If you do have questions for Bill, please use the chat box, and I will be monitoring this box and alerting Bill of any questions at the end of his presentation. I'm going to go ahead and turn off someone's audio. Okay, there we go. And also in regards to chat, we will be recording this presentation and the chat conversation to share with our online presence after today. So please be mindful of a positive presence when communicating in our chat box. So now it is with great pleasure to introduce Bill Bakaitis, Professor Emeritus from SUNY Dutchess. He shares with us today his experience and knowledge from over 38 years of teaching mycology. In addition to his teaching, he has worked with poison control for 30 years and has founded the Mid-Hudson Mycological Association. Bill has delivered lectures, taught courses, and conducted workshops all over the Mid-Hudson region, and we are so thrilled to have Bill with us today. So welcome, Bill. I'm going to go ahead and turn off my video and audio, and I'm going to let you take it away.
Okay. Okay, well, thank you very much. And I'm prompted right before we begin just to mention something about false hellebore. I just received, I worked with poison control for 35 years or so. And normally this time of year, there are some cases where people eat hellebore, false hellebore. They think it's ramps, and it's pretty serious. You could die from it. And they get a number of cases. This year, I think the caseload has tripled, and they're finding that people are staying in the hospital for some time. They're giving heart medication. So please, before you go out and try to collect ramps, know what you're doing. It's not an area for the curious, unless you know what you're doing. Well, welcome, curious naturalists, and I imagine a whole lot of mycological people out there too. This is a program that is meant to be introductory. It's not a high-level program, although some high-level concepts will be introduced. Just probably to start by talking about a good reference you can have. If you don't have the National Audubon Society a Field Guide to Mushrooms, I would recommend that as a field guide when you're going out in the field, the woods, to look at mushrooms. There are 3 taxonomic schemes that are currently in use. Morphology is the shape of the mushroom. You know, it's what it looks like. Does it have stems? Does it have gills? Is it hard? Is it soft? What does it smell like? Things of that sort. Those are all morphology, things which pass through our senses, and we're able then to make a description of mushrooms. There are some more technical things where you do mating, usually on a Petri dish in a laboratory, and DNA analysis, and that's very complicated work there. So these always do not agree with one another. We're not going to spend too much time talking about that today. Just to know that there are these differences. We'll glance at that from time to time as we go through this, but there we go. So I'm normally present this as a walk-in talk where people are right in front of me. I can see your heads, I can see your smiles, I see when you are shaking your heads and nodding your heads, and we can chat. This is a different structure, so I'm just going to treat this as a monologue and talk into my computer here. We had scheduled 4 classes. This is the first of those 4. It's going to be on the biology and life cycles of fungi, and if it goes well, we'll see what happens after that. So fungi, as you probably know, occupy their own separate kingdom. They're not plants of the higher kingdoms, plants, animals, and fungi. They're not plants. you know, and they're certainly not animals, although many of them have animal-like characteristics, and for a long time they have been described as plants. So it's a rather separate kingdom with their own biology, their own life cycles, and some of these get really interesting, as we'll see. Let's just start moving along here and look at some biological characteristics. First thing, it's obvious that mushrooms are not plants. We can see this today. If I were to put on a picture of a plant, you see, first of all, it's green. There's chlorophyll in the plants, and the plants reach up their little hands and they catch the sunshine that's coming down, and through that magic chemistry there, they take carbon dioxide, they spit back out the oxygen, and the carbon they keep. And that's what makes life possible on Earth, that basic process there. They capture about 10% of the solar radiation coming in, so they're often called the first trophic level, energy level. A lot of you will know what this plant is right there. It's wild spinach or Chenopodium, lamb's quarter. Really an excellent spring green. Probably the best one you're going to get. So there you go. So plants just convert solar radiation into carbon compounds, and that gives us the base for life on Earth. Well, you and I get our energy from eating plants. Maybe we don't think of maybe as plants, but the Cheerios you ate for breakfast or the cornflakes were plants, you know. And the eggs, well, the eggs came from chickens which ate the corn or the wheat to make the eggs. So we get all of our energy from plants, even inside of our gut. It's fungi in there which are taking what we have put down there. You know, essentially the alimentary canal is just a hole through our body. It's got chemicals and acids and lots and lots and lots of stuff living down there. I've heard it said that there are more, there are more species of fungi in your gut than there are cells in your body. Now, I don't know how in the heck that can happen, but there you have it. At any rate, there's a lot going on there. Growing up on a farm, I remember when you had a calf, one of the first things you did was reach inside the cow's mouth and take some of the cud and put it in the calf's mouth so that the calf would have all the fungi it's necessary to begin the digestion of food there. Another really important difference between plants and fungi is that plants have seeds. Fungi don't. So when you have a seed, what you've got here is a heavy coat which protects it from the weather, and then you've got a lot of starch compounds in there, energy compounds, and then it's all encasing this embryo. And if you put the seed out, it swells, it bursts. Some seeds you have to nick to help them do that, but then what comes out is the plant. You know, all these little leaf cells are in there. They were already inside there. You know, these little root hairs, this root, they were already inside there, just in an embryonic stage. And so we're just releasing that. The water's releasing it, it's swelling it up, and they're growing. That's quite different when you look at fungi. The reproductive cells of fungi are called spores, and when you look at a spore, there ain't nothing in there. It's just transparent. You know, it's filled with some fluid, but there's nothing in there like a plant. So these are spores, and when this is about 1,000 power, about the size of a red blood cell, say 5 or 10 microns. When when they reach a suitable surface, they just germinate and they turn into a hypha. So this is what we probably in an earlier time, these are called the roots of a mushroom, but this is really the mushroom itself. What we see outside as a fruiting body is just like the apple is to an apple tree. It's with a fruit that produces the seeds. So the spore germinates and turns into this hypha. It's a narrow tube in which it contains the nucleus. There's a cell wall here where one cell can pass the nucleus on to the next and the next. Sometimes they're going to have this, it's called a clamp connection, and they also have a very small pore here called a dolichopore opening, which allows the material to pass from one cell to the other. But that's a hypha. Now, so you have a spore. Here's a mushroom, right? Can't tell it's a mushroom. guild mushroom. There are going to be spores that it releases when it's mature. And here I've diagrammed those as a black spore and a red spore. There'll be more about that later. But this is the haploid stage. It's only like your mother or father's spores. You know, this is half of the entire nucleic material of the genetic material of the mushroom. It just splits into there. And so you got, here's a spore, it just falls down and germinates. Now, because it ain't got no starch in it, there's not much in there, it's got to start earning its living very early. It's got to start eating early. And the way that this is going to do that, it's going to release enzymes from its body out into what we call the substrate, whatever it's growing on, and it's going to digest its food outside of its cell body. And that's quite different from you and I. You know, we put food inside of our mouth, inside of our body to digest food. Fungi digest food outside their body, and one of the things that turns into, and when you look at it at a larger scope, is they are absolutely excellent recyclers. This is one of their native functions. They just release these enzymes and they break down the carbonaceous material in the environment. And by doing that, they turn the carbon back into carbon dioxide, you know, which is necessary for trees and grasses and everything else. And that's how they get their energy. So we've already hinted here now on one of the two major feeding strategies of fungi. Some fungi are saprophytic. They're saprobes, and what they do is they break down organic matter in the soil. They break down the leaves or the wood or the grasses, and that's what they do. They just release their enzymes and they digest and break down that in the environment. Other fungi are called mycorrhizal, and what they do is they send their little hyphae, those little strands, into the root hairs of a cooperating plant. And there they form what's called a symbiotic relationship, a marriage, so to speak, and each shares bits and pieces of its own biology with the other. So it's a very interesting structure there. It's called a mycorrhizal structure. All told, about 90–95% of all green-growing plants form mycorrhizal connections with fungi. Not all the fungi make mushrooms. A lot of the fungi don't make mushrooms at all. But 95% of all plants or so, 85, 90, 95% will form mycorrhizal connections. And so there's a fungus somewhere making that plant grow. So I hope to talk more about that later. If we have another talk in this series later, we'll talk about the ecological functions of fungi, and really this becomes so important in things like climate change. These two ingredients here put CO2 in the environment, take CO2 out of the environment. They're just really important regulators of global climate. All right, so here's a good question. If the mushroom feeds on carbon-based plant material, why doesn't it self-destruct? If it's releasing enzymes which break down carbon and carbonaceous material, why doesn't it self-destruct in the process? I could let you think about that, but because we want to get through this whole program, I'll give you the answer. It's because the cell walls of the fungi are made of chitin and not cellulose. If you've never heard of that chitin, that term chitin, you've probably dealt with it in your life. If you've ever cracked open a lobster claw, that's chitin. Shrimp shells are chitin. So mushrooms are made of the same material that shrimp shells and lobster claws are made of. And that's one of the reasons why mushrooms are difficult to digest. They're not the easiest things to eat, and some people will eat mushrooms, you know, and have a hard time passing them through, and they think the mushroom is toxic. And really, all it is just that it's just a hard thing to digest. This one is a toxic mushroom, though. This is a Tylophosphalius. Phallus means false, deceiving in a sense. So looks like an edible sep, but it's not. So here we are, one of our definitions of fungi. Fungi don't have chlorophyll. They reproduce by spores, not by seeds. They have cell walls made of chitin, not of carbon. I should say they are carbon, not of, what's the name now escapes me. What's most stuff made of? It'll come around. They feed outside of their bodies, and they have distinct sexual characteristics, okay? So cellulose is the word I was thinking of. Their cell walls are not made of cellulose, but of chitin. So that's one of the ways you can define fungi. Okay, let's move on then. Let's look at the 5 kingdoms a bit and talk about evolution of life here on the planet. So we've got fungi as one of the higher kingdoms, plants, fungi, and animals. And then you've got things like algae and protozoa, and then you've got things that are called prokaryotes. Karyot would be like the nut in Greek. That would be the genetic material, the DNA. So you've got certain kinds of bacteria and cyanobacteria that really don't have kind of full-fledged DNA. That their sequence is more like RNA. And then you got protista, and from that we have fungi, plants, and animals. It's been assumed that all of these life forms come from a common ancestor. Long, long ago and far, far away, long ago and far away, there was a common ancestor, and from that common ancestor, we all developed. So here you have the Monera, the bacteria and cyanobacteria, and then the protista, and then you've got animals coming out of that, and fungi coming out of that, and plants coming out of that. And this then starts to define what's commonly called the tree of life. And this can get very specific when you look at the DNA. You can really break down very fine distinctions. And this is the descent of the central dogma of Crick and Watson, that the protein that's in our body came from the DNA, which came from the RNA, which came from the DNA, which came from a common ancestor long, long ago. About 20 years ago, however, actually maybe 40 years ago, 1983. In 1983, Barbara McClintock won the Nobel Prize. She won the Nobel Prize for work she did in the 1940s and the 1950s. And what she was doing there was she was looking at corn, zea mays, you know, the kind of corn we eat, and we make popcorn out of it and feed the cows and all that sort of stuff. She was looking at that and she said, you know, there's a strange thing going on here. There's things happening in corn, genetic things, genetic events which happen in corn, which do not come from the ancestral corn, which didn't come from their mother or their father, from the ovary or the pollen. What's going on here? And as she started to publish this, she was scoffed at. People didn't believe it. And I think part of the reason they didn't believe it, it was odd. She was the first one to really call attention to this, but I think because she was a woman, you know, and women were not given much credence as much as they are today at that time. So no one followed her. She just was pushed to the sidelines. And then in 1983, she was given the Nobel Prize for this. And what she had talked about were jumping genes or transposons. We now call horizontal gene transfer. And particularly when we looked at retroviruses and RNA, and how you could get a virus that could go back and change the DNA, and that came really to be very important when we were talking about HIV and AIDS in the 80s. So her work was just tremendously important, and now that kind of work is used all over the place to do gene editing and to create. Well, we understand how it is that COVID-19 came about. You know, you've got a gene in a bat that gets transferred over to pangolin that gets transferred to a human. You know, well, we now know that is that happens. So if you're not aware of this kind of process, here's one source that you can go to. You can certainly go to the internet and find a whole lot about transposons. So one of the things we now know about fungi, which is right here in the eukarya, the true organisms that have a gene, genetic material that are transferred back and forth, is that there's a lot, it's not nearly as clean as we thought it was. There's a lot of complexity involved in this. And that is going to be a continuing theme in this talk. Mushrooms are incredibly complex. Fungi are incredibly complex. We want to straighten things out. I got to get rid of this stink bug here. There it goes. We want to straighten things out and make things simple. That's just part of our nature. We want things to be simple. But just like the reflection of this forest in the pond, there are no straight lines in nature. There's lots and lots of gradations and a lot of wiggly surfaces. Consider, for example, the slime mold. Now, slime molds are one of those very interesting critters out there that spend part of their life like an animal. I'm going to show you some pictures of them. Like an animal. They're like an amoeba, and they're just coursing through the ground or a log or some substrate. They're moving. They move about a millimeter an hour. You can actually see it with a 10-power hand lens. You can watch it if you just pay attention to it. It's good for mindfulness or meditation, just to go, like watch a celery seed grow. Watch one of these guys move around. When conditions get tough and it's hard for them to continue to make a living, they then pull themselves up out of an amoebic stage, and they develop this plasmodium, and then they make fungal-like spores, and they release the spores in the atmosphere to get from one log to another log or whatnot. We now think that classify slime molds in kingdom Protista. They were called for a long time myxomycetes, kind of fungi, and indeed they are prevalent all over the world. We see them, and they're treated like fungi, not only by people who go out in nature, but by people who treat them. So here's one right here. You can see it's a slimy stuff, you know, it's just moving through this log, and it's moving around searching for food. And here's another one. You see it just moving and streaming around here, and now it's starting to run into trouble, and it says, oops, I got to pull myself up and make some spores, and starting to make some spores. And here it is making these tall stalks now out of the slime. It's just streaming. You watch, it's just streaming out here. It's pulling itself up, and it's making this kind of structure. And here's another one here. This Corpus Fragilis coming up is making a structure. Now, you can't tell the species by looking right here, not by morphology. You can tell, though, by looking at the spore-bearing structure, and they're all different. So this is Corpus Fragilis, one of the ones we commonly see, images like this will be in field guides. That's what you're looking at. Look like they might be wasp eggs or something, but it's really a slime mold. Here's another one, Ceridio mixa fruticulosa, and this is in the stage which is called a poroid stage. It looks like pores, right? And here's a stage which you might call an intermediate stage. You know, you might think, well, maybe it's just growing out here. And here's one called the coralloid form. You might say, oh, well, this is just an older form. But here's the slime right there. It just comes out full blown in this coralloid form, you know, or the intermediate form, or a poroid form. So already we're seeing a kind of complexity where what you're looking at is not exactly what's going on. This is one which is very easy to identify. This is wolf's milk fart. I think they call it that. If you Lycogula epidendrum, Lycogula I think means a wolf's milk slime. Oh, sorry, it's all right. There's another one called wolf's fart. I forget which one that is, I guess. But if you take your penknife and you just puncture this, it'll look like toothpaste. You know, some people call it toothpaste slime. So this is the maturing. The pink one here is the maturing organism. This is the spores that are already fully matured and developed. They puff them open as black smoke, which goes and infects other logs. This is one which almost looks like it's a delicacy from a pastry shop, right? Briffeldia maxima. I mean, it just looks like it's covered with sugar. Boy, like a jelly roll, good to eat. And when you cut it open, oh man, look at that poppy seed inside. Yeah, it just looks delicious. I don't know anyone who eats that one, but there it is. I found it rare in this area. I only found it a few times. This one, however, is common. You see it every season, sometimes a dozen or more times. Folio septica is the Latin name for it. In Mexico, it is called Cacadeluna, or excrement of the moon. And it is eaten by some people there. Here you can see the slime coming, and it's pulled itself up into this athelia, or bun-shaped structure. Got that open, it's got the spore mass inside. There's a lot of interesting stories about this one. In the late 1960s, early 70s, there was a lot of crazy stuff happening out west. You know, psilocybin mushrooms and all these weird cults and alien space invaders and everything that psychedelic stuff can put into your head. They were seeing out there. They were seeing, and this thing showed up in Garland, Texas. And they said, "Oh my gosh, you know, it's a thing from outer space. Get out of here with a before I call the cops." And there it is. So what are you going to do with it? You know, well, they called in. First of all, they called in the first defenders. They called in the fire department. They said, "Get rid of this stuff." So the fire department turned their fire hoses on it. It was just in the streets, and it just went all. They just broke it up. It went all around the streets and the gutters and the drain system, and where it started to reproduce. Well, they called in the National Guard to deal with this invader from outer space, and what it turned out to be was just this very common Cacadeluna. Lots of luna. So there. Another example of taxonomic complexity had to do with lichens. Now, lichen is this complex, really hybrid organism that it is some algae, which lives in very close symbiotic relationship with some hypha of a fungi. Almost all are ascomycetes. So here's an ascomycete, which makes a sort of cup-shaped structure, and it incorporates the algae in that so that it feeds on itself in a sense. The algae captures the energy of the sun, which feeds the fungus. Lichens, as you probably know, are one of the first life forms to start breaking down material that's in the environment. Rocks, for example. They come in a variety of forms. This one, cristata, is often called British soldiers because of that red hats or whatever the British soldiers wore there. So this would be a fruticose form, and this would be a filamentous form. If you go down south, you see a lot of these mosses hanging from the trees. This is a crustose form here. This only grows within 100 yards of the ocean, maybe 200 yards, but very close to the ocean in the northeast. Some of these lichens you find growing over in the gunks were called orchils, and they produced a purple dye when they're mixed with urine. So they would gather them and pee on them and put the wool in there and make that. Some of them were, you would eat them, you know, survival food. It tastes horrible. I've tried them, but it does not taste good at all. And they would be something like this this foliose form. So those are the forms of lichen. I don't know much about lichen, but there you go. In a timeline, if the planet started 4.5 billion years ago or thereabouts, you know, give or take a few years, life began, you know, took a while to get life to begin. All that stuff cooking, but it began, and then our last common ancestor might have been 3.5 billion years ago, and then photosynthesis came. First fungi, 1.5 billion years ago. These were the slime molds. And then increasingly the plant life became much more complex, and algae started, and green plants, and then algae, and then fungi, and then finally animals. So about 500 million years ago, we have land plants and animals and fungi all coming. We think this is a sequence of plants and then fungi and animals came. So fungi are rather recent in this development stage here. Of the entire kingdom, the fifth kingdom, the ones that we're probably most interested in as naturalists going out in the field are the ones that are called basidiomycetes, and those are most of the mushrooms. Time of this chart was made, there might have been 15,000 species. Probably many more than that are identified now. Ascomycetes are things like yeasts and morels, and here they tended to measure genera rather than species. But we know some estimates now are 1.5 million, 2 million species of fungi. Okay. So let's look at here, remind us that there's morphology is not the same as mating, is not the same as the DNA barcode. So there's different ways that you can organize this material, but we're just going to look at the way that they look, the morphology here. So the morphology here is you got, well, slime molds we talked about. You got molds where they have spores that swim around. And you got spores that are airborne, and that's what we're going to be talking about here. The two kinds, mostly the spores that are formed on clubs, and those are called basidiomycetes, and those are these kind down here, and spores that are formed in sacs, and those are called ascomycetes, and those would be these right through here. A morel is an ascomycete, a puffball is a basidiomycete. So we'll move on and have a look at some of these things and look at the life cycles of higher fungi. This signature slide here, this is Amanita muscaria, variety Formosa, except that Amanita muscaria doesn't grow in North America, so they call this Amanita muscaria. So the names of mushrooms change just about as often as the score does in a basketball game. So you can drive yourself crazy in trying to keep track of the names, but this is Amanita muscaria as far as most of us are concerned. So sexuality in the basidiomycetes, and isn't it interesting that we have a phallus mushroom in order to introduce this section here. This is called phallus for the obvious reason. This is one of the ones that looks like a dog phallus. Okay, but these basidiomycetes are going to release their spores on clubs. We'll have a look at some of those clubs in a minute. We saw this mushroom here, releases its spores and it germinates. Now, to complete the life cycle, it's got to find a mate. And so the spore germinates and it goes along and it meets another one. Let's say one of these black spores, we might call it a male, but in mycology they call it a plus. We'll meet one that's a red spore that we might call a female, but in mycology they call it a minus. So a plus and a minus meet and they recognize each other, they're the same species, so they initiate a sex act. That is that they join juices. We call that plasmogamy. They fuse and they now have an organism which is called dikaryotic. They have both the male and the female, the plus and the minus nuclei going side by side in the cells right along for most of the life cycle of a fungus. It's dikaryotic. It's not the haploid number. It's not the diploid number. It is the dikaryotic. They're traveling along as fellow travelers in this game of life, but they don't consummate the sex act. They're just getting to know each other. So this goes on for a long, long, long time. I don't know the age. If I were in a class, I would sit and see if there were children here. I would not go on, and I probably shouldn't go on with much more of that. But you can play with that in your own mind should you wish. Here's something else to consider. The fact that you have two of these hypha joining does not preclude a third, a fourth, or a millionth from joining. So what you've got here is a mass of plasmogamy going on. I won't play with any of these words, but you adults can play with them. What's going on here? You got this incredible mass of this sexual union going on here for a long time. So this is a dikaryotic mycelium here. It's plasmogamy without karyogamy. It's just going on and on. It makes this mycelial mat. But by the time you get a number of them together, we don't call them hyphae anymore. We call them mycelia. And it makes this mycelial mat, and it gets very thick, you know. And if you were to buy some of this, you would call it the spawn. And here it is. You would just see it. You lift up some leaf litter, and you see it binding all those leaves together. And that's the dikaryotic mushroom. That's the dikaryotic mushroom right there.
Hey, Bill, I just want to let you know that we're at 1:37, just so you know.
Okay, thank you. So here it is that this mycelium is going to go up and make this this dikaryotic mushroom. This is Stropharia rugosa annulata. If you want to go to leslieland.com, there are complete instructions how to grow that there. That's what it looks like. And this is a dikaryotic structure up until it gets inside the gill, where it becomes the karyogamy takes place, the sexual act is consummated, and what happens here is that the nuclei fuse, and then they go on and make spores, and here's the spore mass right here. Okay. So that takes place inside of the hymenium. That's the gills on this mushroom here. And that makes this. Okay, this is what a basidia looks like a club-shaped structure. The spores, there are 4 of them. They're on the ends of these structures called sterigma. And there are many different shapes and textures to the spores there. So this prolonged dikaryotic mycelium, multiple matings and parents, makes for a very rich kind of genetic background that the mushrooms have, and a great deal of genetic and adaptive variation that goes on there. And one of the reasons why they've got such integrating characteristics. Now, buckle your necktie or hang on to your hat or something here. If that freaked you out, think about this. This is Schizophyllum commune. You know, we just talked about 2 sexes, plus and minus. Schizophyllum has 28,000 sexes. I don't even know what that means. But you got gender identification problems. Think about this guy right here. Okay, they have 2 sexual loci, A with 300 alleles and B with 90, so over 90. So that's how you get to 28,000. Ascomycete life cycles are similarly complex, even more so. We're now dealing with the complexity of fungi. So ascomycetes make their spores inside of an ascus. Here's one right here. Here's another one. And they generally have 8 spores inside of an ascus rather than 4 spores on Basidia. They have the normal kind of dikaryotic life cycle that goes on right here, this dikaryotic cycle. But they also have another cycle where it's just the haploid cycle, and the male meets the male and they join and they make their own ascoscarp, or their own organism or their own tissue, and the female makes only the female and they make their own tissue, and they can have the male and female, the 2 ones here, or they can have the 2 + 2 + a million more and join in there. And so that's the really complicated sexual cycle in ascomycetes. And if that's not complicated enough, then you also get this asexual cycle where this mold just goes out and it's on its own, and it's having its own asexual cycle, and it can join into the sexual cycle at any time it wants to, or I don't know how you even describe this in any sense, but this is joined in on an annual basis. So it leads to what we certainly call polymorphism, where you can find a wide variety of shapes and sizes of mushrooms, even if they're growing from the same cluster. These are black morels that are, I don't think you're going to find any more black morels out seasons past on those. You'll find blonde morels. And then this guy here, you know, it came from the same group, but you know, you and I would say morphologically, it's not related to these. From a major technical review of morels, we find this language. Each morel may be composed of a variable mixture of mono, male only, di, male and female, multiple, many males, many females together, or heterokaryotic, many males, females, and what is this other thing? And the pairing of haploid nuclei can occur anywhere in the life cycle. Such colonies should be considered populations of nuclei or genes rather than as individual morels. Now, isn't that creepy? You know, I mean, how do you even wrap your head around that? So this inherent genetic diversity can be augmented by the new genetic material from those molds on a seasonal basis. No two morels ever analyzed were found to be genetically identical, even if growing from the same cluster. So interesting stuff. Another layer of complexity. This is the fruiting structure. Here we have a stomach rather than pits on a morel, but this is the fruiting body, and this is the mold that goes along with it. It's called the anamorph. It's the imperfect stage. Well, you can have many different, each of these molds has its own structure going along with it. Constantella cristata, for example, is the same as Morchella genetically. Gives you a morel, although it doesn't look anything like a morel. It looks like a mold. Even more complex, you can have synanamorphs, many kinds of multiple sexual forms over here, asexual forms here. Then you get things where you have, when we're looking at things, here's a mold on top of an Ascomycete mold on top of a Basidiomycete fruiting body. Then you get things like this, where the fungus will have two different shapes. This is the cedar apple. On juniper, it looks like an apple. And it's already happened around here. A couple of weeks ago, we had rain, and those apples send out these long sexual structures. They release their spores, and the spores went on to infect the leaves of apple trees that were out at the time, and they make these little structures that looks like a juniper on it. So this is cedar apple rust, and one of the ways you want to control rust in your apple fields is you cut down all the juniperi around it to make sure that they don't spread their spores. So I'll share one more of these examples, talk about complexity. This is barberry and wheat rust. It's really important because you look at how important wheat is in our diet. You know, it's just everywhere. So this is Pachinogrammus, and has two hosts in multiple forms. This is barberry, as you know, Japanese barberry, European barberry. They come around, and turkeys love these things. I've seen turkeys on top. He's just gobbling those berries up, boy. It's called the obligate host. So you need to have the barberry, and there's a spore on the ground. It's a Basidiomycete spore. That's complicated. It was an Ascomycete spore that then develops Basidiomycete spores, which then infect the barberry. And on the barberry, there's got a process where there's spores on top. They shuffle the genes, the spores on the, and they're sporing bodies on the bottom. They shuffle the genes. It goes to the wheat. The wheat shuffle the genes. They can go back, shuffle the genes, and they go over here to resist the spores. It comes in here. So you've got this you got two things. You got wheat and you got barberry, and you got multiple sexual forms going on here. And this is the pycnium on the top, the aciums on the bottom. You see little aci in there where all these spores are being produced. This is the rust on the wheat. This becomes a short cycle and go back and infect wheat more, can infect the barberry more. But it's going to overwinter, produce a later darker stage, a resistant teleospore. And that then lies on the ground, and it turns into a basidiospore, and that then infects the barberry again next spring. So here you've got a plant, as a fungus, which is able to shuffle the genes once, twice, 3, 4, 5, maybe 6 or 7 times there, another time here, another time there. So at least 7 times in the life cycle of this barberry wheat rust, where the genes get shuffled. Well, when that goes on, you can have rapid genetic evolution. So when you're trying to manufacture chemicals, say, to kill the rust because it's killing the wheat, you're going to lose. And you're going to lose because the rust is going to evolve faster than you can produce chemicals to kill it. And that's exactly what happened with a strain of wheat rust called UG99. I think it stands for Uganda 99, which is when it was discovered in 1999 in Uganda, which is currently threatening to destroy the world's wheat crops. It's really difficult to control that by mass agricultural practices. So there it is. So here's a summing up sort of slide. That's complexity in the fifth kingdom. We got blurred lines between kingdoms. We got slime molds and lichens and transposons. There's unique sexual and reproductive strategies. There are multiple spore types. Some are motile, they swim around. Some are non-motile. Some are resistant. There are kind of mating types. There are sexes. You got bi, tetra, multipolar. You got a dikaryotic mycelium. You got multiple parents, vast genetic storehouses. There's sexual and asexual reproduction. There are anamorphs and synanamorphs and teleomorphs and budding, which we didn't talk about budding. It's where a yeast cell just makes a daughter yeast cell by budding. You got mycelia sterilia, which filamentous mycelia cells will do in the soil, just like budding does. You got alternating generations of forms. You got alternating host forms and spore characteristics. Heterophalic and homothallic mating. You can mate with yourself or you can mate with someone else. So this is an incredibly interesting and complex world out there that's the fifth kingdom. And one of the things that it bears upon when you and I, as curious naturalists, will go out and we look at something in the woods and we say, what is this? Well, that's why they're so often so difficult to identify. So had this been a course that went on normally, the next talk we would have looked at spring mushrooms, and then after that we would have looked at ecological functions of fungi, and then we would have looked at toxic fungi and their edible lookalikes. No, they're lookalikes. Toxic fungi and their edible fungi and their toxic lookalikes is the way I presented that. So there it goes. But right now, I think we're ready for some questions. So you can you've sent your questions off to Christy, and how are we fixed for time, Christy? Can't hear you.
Sorry. So we have about 10 more minutes. Is there anyone in the audience that has any questions for Bill? You can go ahead and chat it up in the chat box. So David, Bill said, what's the difference between a sexual union and consummation?
Well, usually in humans, it takes place almost immediately. In animals, it does. You know, where the sperm meets the egg, and when the sperm, as soon as the sperm penetrates the egg, very shortly after that, there is the joining of the nuclear material. But within mushrooms, it's in two stages. You have plasmogamy, where the juices, the plasma join, they fuse. So in a sense, the sperm would enter the egg, but the nuclei don't join at that point. They stay separate for most of the life cycle of the fungus. And it's only when the nuclear material joins that you have the full consummation of the sex act at that point. I don't know if that answers your question, Dave. Can you ask it in your own words if it doesn't?
I don't see any responses. We have another, oh, he said it was fine. Another question that was asked is, what is rust exactly? And simply, if possible, can extend organic farming alleviate its problems for crops?
Yeah, well, rust is a fungus. It is essentially the mold state of a fungus. And it is usually treated by chemical means. If you want to deal with this in an organic way, if apples are one of the ways you can think of this. Most apple orchards around here are going to spray because we want to have apples that have a smooth, perfect skin. We don't want them to have that apple scab on them, for example. So so we spray for that. One of the ways to control some of these things is when there's an alternate host, is you cut down the alternate host. So you would cut down the alternate host of cedar apple rust by getting rid of all the cedar trees around. It's not so much a problem with pears because pears have a russet skin anyway. There are some strains which are resistant. Rust so it's a path. It's not one of my areas of specialty at all. I don't know if I have much more to say about it than that.
My question, Bill, is for some of the mushrooms that you had talked about, like the cedar apple. Yes. Does that really affect the juniper tree when it's growing? Does it, inhibit anything? Is it weakening the immunity of that plant? Is it just kind of like the growing body for which it grows on? Can you explain more? Does it affect in any other way?
Sure. A parasite, and these are parasites, a good parasite never kills its host. If it kills its host, it's dead. So it can weaken the tree. And some trees become weakened more than others. I have an espalier where I have three organic or heirloom apple types on it. One is a pippin, one is a Zabergal, and one is a wealthy apple. And the wealthy apple is just, I mean, if a pest just looks at the tree, it seems to get the disease and just makes huge apples. Apples are as big as grapefruits, huge, beautiful apples, but it's very, very susceptible. So there's no way I can grow that tree without spraying it. I just can't. It just won't do. And but on the other hand, there are a lot of wild trees around, that have survived, and they're tough. You throw everything at them and they still survive. And so plants vary in their regard to throw off those kinds of disturbances. But it is a biological disturbance when you have a pathogen, and it has to, if it has a host plant that's live on, if it kills the host, it dies too.
Okay, thank you. We do have one other question of when you said that plasmogamy could go on for a very long time. Can you explain more? Are you talking like months, years, decades? What does that mean by long time?
Yeah, certainly months, certainly years. If you have, when you pick a mushroom, the mycelium stays in the ground. So there are some fungi which go on to live for, generations. They just, for 100 years or so. And within that, it is, well, we're getting very technical here, but and the example I'm thinking of really probably doesn't have a dikaryotic mycelium, but many of them just have, they're perennial mushrooms. They live, say a mushroom on a tree, a tree that like Trichaptum biformis, or say a turkey tail mushroom on a tree. It's going to, it'll grow, it'll fruit now, it'll fruit next year, it'll fruit the year after that. It's the same mycelium inside the tree, which is growing. So that's all dikaryotic inside the tree. So certainly for months and years, some things like a puffball is going to be an annual thing. It'll grow, make a puffball, and then the spores will make a new one next year. But some fungi live for years, for decades, and so that would be a long time there. Yeah.
Okay, wonderful. We do have one last question that's been asked, saying what percentage of fungi form our mushrooms? What percentage of fungi form? What percentage of fungi form mushrooms?
Yeah. maybe 10%.
Oh, wow.
Yeah, yeah, maybe even less than that. About in the forest mushroom, I mean, that's you're talking here about ectomycorrhizal fungi and saprophytic fungi combined. So maybe 10 or 15% of fungi will, maybe even less than that. You know, around here there are about 3,000 or 5,000 species of fungi, mushrooms that we see in the field. You know, no one knows them all. There's so many of them. So if they have 3,000 species of mushrooms around here, you've got 1.5 million species of fungi. Well, you know, you're down to hardly any of them form mushrooms.
Are there any other questions for Bill today? If not, thank you so much. This concludes our Mohonk Preserve's first online public program. We want to thank you all for joining us on our first virtual public program presentation. Please be on the lookout for an evaluation in your emails. Also, if you're in the position to make a donation to the Mohonk Preserve, we would greatly appreciate your support as we continue to provide programs for our public and keep our operations moving forward during these difficult times. Your charitable donation would mean a lot to us. So I have provided a link here in our chat, but thank you so much for joining us. Bill, I have enjoyed working with you over the past few days, weeks, getting this webinar up and running, and hopefully we can present more webinars on mushrooms and hope to see you all in future programs, events, and on our trails. So thank you so much, everyone, and thank you, Bill.
Oh, goody. Thank you. Right. Thanks very much. Right.
Yeah.
Bye-bye.
Bye, everyone. Thank you.